Welcome to the PubMed Daily Review for April 30th, 2026. Today we scanned 2,897 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Neurological Disorders and AI. Investigating neurological disorders through diverse biological and computational approaches, including AI and robotics. This cluster covers 346 papers. Representative work includes clinical and electroencephalogram characteristics of pilomotor seizures, a qualitative exploration of ethical aspects of using AI in Parkinson's disease, patient panel study, and effects of a soft robotic exoskeleton for gait training on clinical outcomes in patients with Parkinson's disease, randomized controlled pilot study. Trend 2 of 5, Molecular Biology and Health. These papers explore molecular mechanisms underlying health, disease, and organismal development across diverse species. This cluster covers 672 papers. Representative work includes heterogeneity of treatment effect, what baseline prognostic factors tell us about immunotherapy benefit, single-cell profiling unveils key regulators of skeletal stem cells in chicken and human embryonic limb development, and phenolic compound from Arthrosper platensis and Chlorella vulgaris enhance growth, digestive function, antioxidant capacity, and immune-related gene expression in Nile tilapia. Trend 3 of 5, Digital Health and Interventions. These papers explore digital tools and data-driven approaches to improve health outcomes and interventions across diverse populations. This cluster covers 527 papers. Representative work includes sexual assault prevention via bystander intervention using Instagram Reels as a communication channel. Experimental design study. Benefit of the N of one approach versus aggregate analysis in tracking individual trajectories during pregnancy. Comparison of longitudinal wearable observational studies. And building EVA, education, vinculation. Why Auto-Ayuda tutorial for the development of a digital mental health chatbot for adolescents living with HIV. Trend 4 of 5, Ecology, Language and Materials. This trend explores how ecological systems, linguistic structures, and material properties interact and are analyzed through computational and scientific methods. This cluster covers 492 papers. Representative work includes B. Hymenoptera, bycatch assemblages collected in bull traps at interfaces of cotton agroecosystems in Alabama, spatial distribution patterns and causal mechanisms of rainfall-induced cluster landslides, a case study from southwestern Yunnan province, China, and syntactic language change in English and German, metrics, parsers, and convergences. Trend 5 of 5, Medical Imaging and AI. These papers explore AI's role in analyzing medical images and clinical data for diagnosis, prediction, and treatment assessment. This cluster covers 860 papers. Representative work includes quantitative muscle magnetic resonance imaging demonstrates resolution of muscle edema in refractory dermatomyositis. A post-hoc analysis of a proof-of-concept clinical trial of Surufatinib, data-efficient language model for assessing pulmonary embolism diagnostic certainty from radiology reports, model development and validation study, and predicting pediatric urological surgery duration through multimodal patient-physician feature fusion, deep learning, framework incorporating clinical text embedding. Up next, our first paper out of 5. Multimodal framework for the joint analysis of single-cell RNA and T-cell receptor sequencing data predicts T-cell response to cancer immunotherapy by Chu-Jun He and Caroline Uhler from Wendy Schmidt Center, Broad Institute of MIT and Harvard, Cambridge, Massachusetts, USA. Let's break down its key contributions. T-cells play a critical role in how well cancer patients respond to immunotherapy. Recent advances allow us to look at both what T-cells are doing, their RNA, and what they recognize, their T-cell receptor, at the same time for individual cells. This opens up exciting possibilities for predicting treatment success. Our research presents a new computational framework designed to unlock the potential of this combined data. We developed a new method called TRIM, which stands for TCR-RNA Integrating Model. TRIM is a sophisticated computational framework that uses a type of machine learning called a multimodal variational autoencoder. It's designed to take the complex information from both RNA sequencing and T-cell receptor sequencing from individual T-cells and integrate them. TRIM learns a shared pattern within this data, taking into account factors like the patient, where the sample came from, and when it was collected, to predict important T-cell characteristics. We tested TRIM on 3 different patient datasets, including individuals with head and neck cancer, colorectal cancer, and a broader pan-cancer group. The results were very promising. TRIM was able to accurately predict key aspects of T-cell behavior, specifically their clonal expansion and their transcriptional states. Importantly, it could make these predictions using T-cells collected from blood or normal tissue before treatment began, showing its potential for early assessment. This study demonstrates the power of integrating single-cell RNA and TCR data using our TRIM framework. Its ability to predict T-cell responses from early samples is particularly exciting, potentially allowing us to identify patients who will benefit most from immunotherapy. While these findings are significant, further research in larger patient groups will be essential to fully translate this into routine clinical practice. Up next, our second paper out of 5, Methylation entropy as a novel dimension in liquid biopsy, enhanced multimodal framework for cancer detection and tissue of origin classification by Xiaoqiang Jia and Xiaoyang Han from University Cancer Hospital, Inner Mongolia Campus, /affiliated Cancer Hospital of Inner Mongolia Medical University, Hohhot, China. Let's break down its key contributions. Detecting cancer early and non-invasively is a major goal in oncology. Circulating cell-free DNA, or cfDNA, found in our blood, holds great promise for this. However, current detection methods can be further enhanced. This study explores a novel characteristic of cfDNA called methylation entropy, which measures the variability in DNA methylation patterns to see if it can improve our ability to detect multiple cancer types and pinpoint their origin. To investigate this, researchers developed a specialized panel to analyze cfDNA from over 500 individuals, including cancer patients and healthy controls. They used deep sequencing to measure not only DNA methylation levels, but also fragmentation patterns and, crucially, methylation entropy. This allowed them to build sophisticated models, including one for multi-cancer detection and another for identifying the tissue of origin for detected cancers. The results were highly promising. Methylation entropy alone showed a strong ability to distinguish cancer from non-cancer, achieving an AUC of 0.938. When this was integrated with other cfDNA features into the EMST model, the performance dramatically improved, reaching an AUC of 0.979 with high sensitivity. Furthermore, the tissue of origin model demonstrated impressive accuracy in identifying where the cancer originated. In conclusion, this study highlights methylation entropy as a robust and valuable biomarker for cancer detection. The multimodal approach, combining entropy with other cfDNA characteristics, offers a significant leap forward in non-invasive diagnostics. This enhanced framework holds the potential to enable earlier and more accurate cancer detection, ultimately improving patient management and outcomes. Up next, our third paper out of 5, combination treatment with synthetic gRNA/Cas12a and gRNA/Cas9 ribonucleoproteins disrupts HIV replication and expression by PubMed and GQ. Yao from Center of Excellence in Inflammation, Infectious Disease and Immunity, Quillen College of Medicine, East Tennessee State University, Johnson City, Tennessee, 37614, USA. Let's break down its key contributions. HIV remains a major global health challenge. While treatments can control the virus, they can't eliminate it because the virus integrates into our DNA, forming hidden reservoirs. This means a functional cure, one that truly eradicates HIV, is still out of reach. We need new strategies, like gene editing, to target and disable this integrated viral DNA. To tackle this, researchers used a powerful gene editing system involving synthetic guide RNAs and Cas enzymes, delivered as ribonucleoprotein complexes, or RNPs. They tested these RNPs in T cells infected with HIV. The goal was to see if these genetic tools could specifically target and disable the virus's own DNA, leading to disruption of its replication and expression. They focused on conserved regions of the HIV genome to ensure broad applicability. The results were very promising. Treatment with a specific Cas12a RNP, targeting the NEF/3' LTR genes, reduced HIV P24 protein by about 60% and HIV mRNA by about 70%. Even more impressive, a sequential combination of 2 different RNPs, G4/Cas9 followed by G5/Cas12a, achieved a remarkable 75% reduction in P24 protein. DNA sequencing confirmed that these treatments effectively created disruptive mutations in the viral genome. This study demonstrates a significant step forward in the fight against HIV. The use of synthetic gRNA/Cas RNPs, particularly in combination or sequential treatments, shows strong potential for disrupting HIV replication. By targeting conserved regions of the virus, this approach could be effective against a wide range of HIV strains. While further research is needed, this offers a promising new avenue for developing a functional cure for HIV. Up next, our fourth paper out of 5, Nivolumab plus ipilimumab and cobimetinib in previously treated microsatellite-stable /DNA mismatch repair-proficient metastatic colorectal cancer. The Phase II CheckMate 142 trial by M.J. Overman and M.A. Morse from Department of Gastrointestinal Medical Oncology, University of Texas MD Anderson Cancer Center, Houston, USA. Let's break down its key contributions. Colorectal cancer is a major global health challenge. While immune checkpoint inhibitors have revolutionized treatment for some cancers, their effectiveness in microsatellite-stable /DNA mismatch repair-proficient metastatic colorectal cancer has been limited. Based on promising preclinical data suggesting a synergistic effect between ICIs and MEK inhibitors, this study explored a novel combination therapy in patients with previously treated MSS/PMMR metastatic CRC. This Phase II CheckMate 142 trial enrolled 30 patients with previously treated microsatellite-stable /DNA mismatch repair-proficient metastatic colorectal cancer. They received a combination of Nivolumab, ipilimumab, and cobimetinib. The primary goal was to assess the objective response rate, with a median follow-up of nearly 5 years to understand long-term outcomes and safety. The results were quite modest. The objective response rate was only 3%, with one patient achieving a partial response that lasted over 32 months. Median overall survival was 8.2 months. While the disease control rate was 30%, the treatment-related adverse events were substantial, with 30% of patients experiencing grade 3 to 4 toxicities. In conclusion, this study found that the combination of Nivolumab, ipilimumab, and cobimetinib had limited clinical activity in patients with previously treated MSS/PMMR metastatic colorectal cancer. The observed toxicities were also a significant concern, with a notable percentage of patients experiencing severe adverse events. Therefore, further investigation of this particular regimen in this patient group is not recommended. Up next, our fifth paper out of 5. Targeting GPR34 in damage-associated macrophages enhances anti-tumor immunity and the efficacy of Surufatinib in pancreatic cancer by Xiaofan Guo and Jihue Hao from Center, Tianjin Medical University Cancer Institute and Hospital, National Clinical Research Center for Cancer, State Key Laboratory of Dragability Evaluation and Systematic Translational Medicine, Tianjin Key Laboratory of Digestive Cancer, Tianjin's Clinical Research Center for Cancer, Tianjin. 300060, China. Let's break down its key contributions. Pancreatic cancer is incredibly deadly, partly because of the shield created by immune cells in the tumor. Tumor-associated macrophages, or TAMs, are key players in this shield, making it hard for treatments to work. Surufatinib is a drug that targets these macrophages, but we don't fully understand how it works or why some patients don't respond. To understand this better, the researchers used advanced single-cell sequencing on tumors from patients treated with Surufatinib and chemotherapy. They also used mouse models and lab experiments. They identified a specific type of macrophage called GPR34 plus DAMs that seemed to be linked to treatment resistance. They found that these GPR34 expressing macrophages were much more common in patients who didn't respond to treatment. These macrophages seem to work by releasing certain signals that lead to T cells becoming exhausted and unable to fight the cancer. Excitingly, when they blocked GPR34 in preclinical models, the combination therapy worked much better. This study highlights GPR34 as a promising new target to improve pancreatic cancer treatment. Identifying these specific macrophages could help predict who will benefit from therapy. The next step is to test drugs that block GPR34 in human clinical trials to see if we can improve outcomes for these patients. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.